Okay, so today I'm going to talk about joint work with Adi Livnat, Christos, and Aviad Rubinstein, and Greg Valiant. Um, and so what we thought about is the question of how a trait becomes fixed in a population, a beneficial trait. So and so this question can have many kinds of answers, and sort of the first one that population genetics gives, I think, is that the trait is completely determined by a single gene. By selection, the gene gets fixed, and then the trait also gets fixed. This is at least sort of the first naive explanation. And but we know that many traits depend on multiple genes. And population genetics also tries to account for this. So one way that's normally done is to assume a kind of independent contribution to fitness from each gene. So it might be additive or it might be multiplicative. Or if there are dependencies, there's a simple structure or some kind of special structure assumed about the dependencies between the genes. And one thing that Les pointed out on Monday was that as we know more, our standards for explanation get higher. And I think what we know now, and what biology seems to tell us, is that a trait can be the product, especially many of the traits that we care about, can be the product of a very complex interaction between very many genes. And so what we'd like is an explanation that can use what we know about population genetics without making any assumptions about what we don't know from biology, which is how sometimes multiple genes will come together and produce a trait. And we'd also like a polynomial time explanation. Because the trait may involve many genes. So how do we think about this problem? Well, we consider n genes, which I'll call x1, x2 through xn. And they'll interact in some way, which we may not understand, but at the end of the interaction, either a trait is produced or it's not. So here we'll think of genes as having two alleles each, and so we'll write the setting to be either +1 or -1 depending on the allele. And so what we might know is that for different settings of these genes, a trait will either be produced or not. And we don't understand necessarily how the trait is produced. And what we want to know, the question we want to know, is that if a trait is present in the population, what happens over time? Will the trait become fixed in the population? How long will it take? And and what will the genotypes of the population look like after fixation? So let me describe a concrete example to explain how we thought about this problem. In 1953, Waddington conducted an experiment on a population of flies. And the first thing that he did was he applied heat shock to the young flies. And then he found that some develop a special structure in their wings, which is called cross-veinless. So the wing of the fly will have veins running this way, and normally in the population you also have veins going here. And what he found after applying the heat shock was that in some of the flies these veins weren't present after development. Okay, so then he selected just those flies that satisfied this property, and then he reproduced them to create the next generation. And then he repeated this for many more generations. And so what did he see in the first time he applied the heat shock? Maybe something like 23% of the flies developed these cross-veinless wings. In the second generation, the percentage went up to something like 56%. And within roughly 25 generations, nearly all would develop cross-veinless wings after applying the heat shock. Right? And I guess this doesn't seem so surprising. This is how we get bulldogs from wolves or wherever. But what was surprising was for some of the flies, he didn't apply the heat shock. And some of these flies also developed cross-veinless wings. So whereas before the heat shock was necessary to bring out this feature, it happened without. So one thing that's interesting about this example, I think, is there is no single gene explanation for what happens with the numbers that Waddington got. And 2 years later, Stern gave an informal explanation that uses threshold functions. And I'll try to give you a sort of generalization of this explanation. Okay, so suppose that whether or not you have cross-veinless wings is a function of these n genes, and also whether or not you apply the heat shock. Then you could, so I'll say that this is roughly n over 3, so that your one setting to the genotype will determine, and also whether or not there's heat determines whether or not the feature is present. Does this make sense so far?
So you're saying the heat shock is like way more important than any one gene?
Yeah. At the beginning. So now how do we keep track of the genetics of the population? So we start with what the genotype frequencies of the population, and to compute the next generation. So if this is at time t plus 1. So here G is just the function that's 1 exactly when this condition is satisfied.
Minus 1 otherwise. And what otherwise, sorry?
Oh, sorry. It'll be 1 when the condition is satisfied and 0 otherwise.
0.
Yeah, sorry. Okay.
Xi's are plus or minus 1?
Sorry?
Xi's are 0 1 value?
I would like to take the xi's to be plus 1 minus 1. Yeah. It's just, I think eventually makes the explanation clearer, but it's not too important. We would just switch around the values. And so, this is just the average value of the xi's in the next generation if we had random mating and full recombination. And we take these marginal expectations, and for the next, for the distribution of the next generation, we use a product distribution where each of the expectations of the bits is given by this average. And so after doing the calculations, you find that after enough time, the function is almost always satisfied when you, so this is the variable for heat shock, right? And you also find that the mu i's have gone up. And what this means is that the function will also be satisfied when no heat shock is applied.
Ui is the fraction of flies at this given generation with gene I, for instance?
Sorry, so Ui is the average value of gene Xi over the flies that had the cross-veinless wing feature. Yeah, thanks. Does that make sense? So all we've done is we've accounted for selection, and we've assumed that we're in a product distribution, that we're on this right manifold.
What is that symbol?
This one?
What
sorry,
is the symbol?
Sorry, it's a quarter. Oh, this one?
Oh, that's a quarter. Okay.
Oh, yeah, sorry. And it's also maybe not really a quarter. I just, estimating.
It may or may not be a quarter.
Yeah. It's like a flag. Yeah, okay. Okay, so yeah, I started mentioning this. This is an approximate calculation. One thing we're assuming is an infinite population, right? We're just keeping track of the distributions and not accounting for sampling effects. And we also assume that we can describe each generation by a product distribution. But I'll talk more about those assumptions when I sort of pose the general question and how we address it.
So this is the explanation for the Waddington experiment.
This is like a generalization of it. I mean, flies are, so I've assumed also that the flies are haploid when they're actually diploid. And, Stern's explanation involves actual genes in the flies, and he's trying to get the same numbers. But this is sort of the idea.
Threshold.
How many genes did he actually estimate the number of genes involved?
I think it was like 8. Is this?
Yeah.
Yeah. I could. Yeah. Don't quote me on that. Okay.
And did you find this regardless of the starting distribution? Or the starting?
So in some sense, this should happen regardless of the starting distribution, right? Partly because of the infinite population and the
Away from the boundaries.
yeah. I mean, it's only really a good approximation when the flies are when the condition will hold with some good probability. But it's also, if you just look at the infinite case, it will still happen no matter what. Yeah.
It should still depend upon the number of inputs at which G is 1 only. So that the convergence time should depend upon.
Yeah, no, that's true. And it does, yeah. Okay. Okay, but so this is a specific solution to this particular trait, and I promised that we want to not assume anything about how the genes interact or assume that the function looks like a threshold function. Right? So what we want to consider is arbitrary Boolean function. Oh, yeah, it shouldn't. Okay. Okay, so now we're going to consider arbitrary Boolean functions, and we'll call them f instead. And instead of being 0, 1 value, we'll let f take 2 values, just 1 plus epsilon or 1. So this is because we want to consider weak selection. So the Waddington's experiment is a very harsh sort of type of selection, and we're interested in less aggressive kinds of selection. And so I'm sometimes going to talk about satisfying f, and that just means that it takes value 1 plus epsilon. And what f is going to be the additional fitness that you get by having the trait, right? And otherwise, f of x is going to just be 1. Okay, and so we still we'll be keeping track of a product distribution at each generation. Where the average values of each bit is so now we're just weighting according to this weak selection instead of G. But this is still what the average value would be with random mating and full recombination, starting from if your parents come from this distribution, then this will be the average value in the next generation. And then we're going to assume that the next generation is actually produced by the product distribution that these average values describe. Yeah, so I mentioned, so there's the fact that we're assuming there are product distributions, and Umesh a couple days ago covered this issue, similar issue in his talks. So we'll appeal to Nagalaki's theorem. So we know in the absence of selection, after roughly log n generations, we do move to a product distribution over the marginals. And Nagalaki's theorem says that with weak selection, after enough generations, the process we consider over the right manifold will be a good approximation to the actual process. Okay, so the first thing we can show is that, which is more general than the last thing, is that if f is a monotone function, so a function is monotone, a Boolean function is monotone if when it's satisfied at a point, flipping a bit from a -1 to a 1 means the function has to be satisfied at that point as well. And if the starting probability, so I'll use this for the starting probability. Then in linear time, so after this many generations, the probability that f is satisfied is very close to 1. And so what about general Boolean functions, which is the goal we set out with? So if you think about it, we'll see that with the way we've defined this framework, we can't get this. So the XOR function.
As a parity function, by the way, the same that is troubling all computer
the parity
science
function, which we understand, but seems tough for biology.
For different reasons.
Right, for different.
So for example, if you start on the uniform distribution, right, where all these expectations, the average values are 0, the function will be satisfied with probably half, but in the next generation you won't go anywhere. You'll just stay in the same place.
It's unstable. Right?
It is unstable.
It is unstable, and so that brings us sort of to the next thing we'll think about. So, like I said, this is an approximation, and one of the ways it's an approximation is in that we're assuming an infinite population. And we know that sampling effects are an important sort of part of what happens to populations, and there's fixation without selection. In this case, it turns out that sort of random drift is going to be important for fixation of this beneficial trait. So what we can show with finite populations is for any Boolean function, So we'll take a fixed population size, and they'll both be polynomial n 1 over epsilon and 1 over the starting probability. With almost, with probability almost surely, almost 1, the population all has the trait. Every individual of the population will have the trait.
In here, mu naught, like we're saying, mu naught is from the product distribution, whereas like above, you would get that it would go to it by Nagalaki, but here in finite population, you start there.
This statement, I'll define all the generations, but we're assuming that, at this beginning, this is when you've started from a product distribution, and you're always in a product distribution. I mean, that's what the theorem is proved for. Yeah.
Finite population.
Yeah, it's a population size. Sorry.
If you find population size, what sense are you always in the product distribution? You're in a given sample. You're not, but you're.
Yeah. So.
Doesn't that matter or not?
I think what we're saying is that you're close to it. If you're in a given sample, you're not. But when we produce the next generation, we'll assume you're coming from that product distribution that's given by the frequencies of the finite sample.
So the way you do the recalculation.
Yeah. So let me say a little bit more about the finite population. So we assume if you have a population at time t, can everyone see down here or that wants to? Okay. So we'll start with a product distribution in time t. We'll take a finite sample of size n, and this will give us a new product distribution based on the empirical expectation, and then we use a selection step to produce the next generation distribution, and then you do it again.
So if mu naught is f, mu naught of f is not going to be like tiny as n gets big, there's going to be like a lot of satisfying variable combinations, right?
Yeah. So there are some restrictions to the parameters for this term to hold.
So
you would
evolution.
let you not have tiny, but then you get
So the point of this is that evolution cannot solve satisfiability. Okay?
Yeah.
As a result, you need to have plenty of assignments, so it will be trivial to solve satisfiability.
Mm-hmm.
So only in these cases we have a chance to evolve. I guess that's one way.
Right, but here you said for any f, so I'm kind of confused the class of f's.
Sorry. Okay. So any f, it's Boolean still. What we've assumed about the interactions is that between the genes is that there are two alleles for each gene, and however they're set, we don't really understand whether or not the trait gets produced, but it's determined by that. And so I guess
You are puzzled about unsatisfiable F?
Maybe mu t, measure of the points where f is equal to 1 plus epsilon is equal. I think that's what you mean.
Sorry. So by this I mean a probability. So this probability is 1. So that by the end of this process, every individual, everything satisfies every individual.
F is equal to 1 plus epsilon.
Well, the expectation of f will be 1 plus epsilon. Sorry, it's just bad. Yeah, I'm using this for the density. Yeah.
So there's two values of f of x only.
Yeah, there's only two values. I mean, so we've in order to raise your standards, sometimes you first have to lower them. So we're simplifying in other dimensions, but sort of the complexity of these interactions, at least to a computer scientist, seems quite intimidating, I think.
So this is kind of like the picture that you were advocating, where you've got maybe some very complex interactions, but there's lots of different possible good interactions. I think.
Yeah. And so we do have some restrictions. I mean, the selection right now needs to be less than 1 over n, the strength of the selection, although we believe that even for constant epsilon, this should be true. And the starting probability depends on the population size to some extent. So it has to be substantially more than, say, 1 over the population size.
You saying you're learning parodies somehow? I don't understand.
No, so there's no.
Because they're functioning by parody.
Yeah, so for learning parodies, the idea is that the function could be labeled by an arbitrary parody. You don't know which one it is, and what's difficult is to determine what it is when you see sort of inputs and outputs to the function. That's what's difficult about learning parodies.
I'm mistaking.
So here, we have.
Population only consists of even parodies.
Yeah, this could be the case if f were the parity function. Then, at the end of this process, in fact, so if I go a little bit further, you'll see at the end of this process, all the genes will be fixed, and they'll be fixed in a way that satisfies the function.
One solution.
Yeah. Am I out of time? Okay. So, yeah. So we won't see that, but that's true.
But the way it does it is only find one solution. Because if you have more than one solution, you're screwed.
Yeah. Otherwise, you won't always be satisfied because you'll have the product distribution from those. Yeah. Okay. So I guess if there are any other questions.